This shows here a layer of epithelial cells. Okay, for example, let's think these are lining the colon. Okay, so the colon is lined by this layer of cells, forms a complete layer protecting the inside of the body essentially from the outside where the digested food is and waste and so forth. So these cells are always going to be under sort of mechanically they're going to be removed and pushed about. So imagine if that cell goes there, then the neighboring cells will divide so as to regenerate a new one. New cell there, just enough. But what won't happen, unless there's some kind of disease going on, is that the cells will not divide in an uncontrolled fashion. So if that happened, then what would happen is you'd end up having a layer of cells which is more than one thick. And quite often, if this did happen, the cells would lose some of their typical characteristics, so they'd become just a general sort of circular shape and so on. And this would be the development, this would be a tumor. So cells are dividing in a way in which they're not supposed to. The control has gone. This would probably be classed as a benign tumor because it's not actually doing that much harm there. When it becomes malignant is when the cells somehow manage to escape through this layer, which is called the basement membrane. It's made of connective tissue, and it separates the epithelial cells from the rest of the body. And what I've drawn here is a blood vessel, so a capillary, some red blood cells in it. And if that tumor became malignant rather than benign, the tumor could is managing to grow below the basement membrane. It's broken through the basement membrane, and some of those cells may actually end up getting into the capillaries and spreading throughout the body and seeding in multiple different places, in which case that would be a fully malignant tumor. So this is what's happening in cancer. So in cancer, you have the control over cell division has been lost. And generally speaking, it's lost because there's been mutations, there've been changes to the genes in those cells, which allows them to behave in this way. And it's if you think about most cancers, childhood cancers are much rarer than adult cancers, which typically appear in their people's late 40s and early 50s, where there's been a long time for the cells to be exposed to mutations. Also, most cancers do occur in epithelial tissues, so most common cancers, breast, lung, and colorectal and so forth, it's epithelial cells which are undergoing a lot of division throughout someone's life. So there's lots of chances of things going wrong and accumulating mutations. Okay, so let's talk about the role of tumor suppressor genes, which is it's kind of a little bit misleading, tumor suppressor genes, because what they do is they don't really suppress tumors as such. They control mostly things like cell division, also DNA repair, and those sort of processes, which if they go wrong can lead to cells dividing rapidly out of control and forming tumors. So let's go back to the first tumor suppressor gene which was identified, which is called the Retinoblastoma gene, and that's usually given the initial RB. Right. The Retinoblastoma gene was came up. There's a disease called retinoblastoma. It's a cancer which is unusually occurs in children in the retina. And what happens if you think the inside of the retina is a layer of It's a layer of cells, and the division, they form a single layer, and the division, the controller division, is lost, and tumors start to develop. And these are visible in the children through their pupils. Their pupils look white things through the pupils in their eyes. Now, the retinoblastoma story starts sort of in the mid-80s, and this was the first tumor suppressor gene to be discovered. Now, there are two kinds of retinoblastoma. There's two patterns in which it occurs. Right, there's the one is called the familial, and one is called the sporadic. Now, in the familial one, it obviously runs in families. Those families also have a higher incidence of other cancers. And quite often it occurs in both eyes. So tumors appear in both eyes, around the same time. Whereas in the sporadic version, there's no pattern of inheritance in the family, no familial history of it. It's usually one eye only, and it's well, in the children that get it, obviously you can't say that their families are having increased incidence of cancer, because they're the first people in the family to actually get the disease. So you can't really talk about whether it's associated with higher incidence of other cancers. Now, the reason people got a handle on this was because if you looked at the karyotype, and if you remember what a karyotype is basically, if you get you catch a cell in cell division, and you look at all the chromosomes there, and you arrange them in homologous pairs, and of course, normally you're going to have 23 pairs of chromosomes. And it was found, if you look at the karyotype of the dividing cells in retinoblastoma, not always, but very often, there is a noticeable deletion in chromosome 13. So, if you have your chromosome 13 and there is a little bit of it missing, those are two chromatids, there's a little bit of it missing in patients with this, it enabled geneticists to home in on this section. And I think in 1986, someone managed to clone a gene which encoded an mRNA, and that mRNA was missing in a lot of retinoblastoma tumors. Okay, so the gene was given the name RB, and because they knew what the gene was, they could investigate children who had the familial version, children who had the sporadic version. Now, what they found was, when they looked at the gene, is the familial there's one bad copy of the gene in every cell in the body. Sorry, every cell. That's sometimes they're called somatic cells. So it just means cells in the body. So they've obviously inherited, in the familial thing, they've inherited from their parents a defective copy of that gene. It's not working. Usually it was found that it's not so much a point mutation where you change one base and that leads to one amino acid being substituted. It usually one base gets inserted or deleted, which of course leads to a frame shift mutation, which means only a small portion of the protein the mRNA that's made actually makes any sense, and the rest has all been shifted one along, and the protein made is totally non-functional. So it's kind of like a deletion of the protein. So they found that, and then that in all their somatic cells, okay, the cells in the body, they had one bad copy, but in the retinoblastoma cells, there were two bad copies. So the theory was the reason why these individuals develop the cancer is because they're born with only one copy. Now, when they've got one copy, that's enough to stop the retinoblastoma cancer starting. But if any subsequent mutation occurs in those cells and knocks out the other copy, then they develop the disease. That's in the familial. Let's have a look at the sporadic, and you can probably guess what happens with the sporadic. They had a look at all the somatic cells. The somatic cells all have two good copies. But the retinoblastoma cells, the cancer cells, have got two bad copies. So in these individuals, the sporadic individuals, it's much less likely because they've had two mutation events that actually occur in the retinal cells. One where one copy gets knocked out, and that's not a problem. And then a second event occurs where the second gene is knocked out. And once you've got no copies of that gene, then the cells they're not subject to the normal constraints surrounding cell division, and they seem to divide out of control. So that's how they discovered the retinoblastoma gene. Right, so that's so much so for what happens if you haven't got copies of it and so forth. Oh, and I'll just say in the familial case, the fact that if you've only got one copy in every other cell in the somatic cell, right, in other cells, if you get a mutation in those cells, then they can develop cancer as well. It's not just the retinal cells. So the retinoblastoma gene, although it's called the retinoblastoma gene, is not just confined to having a role in retina cells. It's every cell in the body that can divide. So anyway, the gene was sequenced. They predicted the amino acid sequence, and they looked at the structure of the protein. So what can we say about the RB protein then? Well, first of all, it is incredibly complex in terms of functions. It's got lots of different functions. It binds 100 plus other proteins. Let's call them gene products. So it's related to the way 100 plus other genes work. And if you think about hemoglobin, that's got a very simple function. It does one thing really, and that's bind oxygen. That's all it does. So it's quite easy for biochemists to look at the structure of hemoglobin and say, yeah, that's what it does, and characterize it. With retinoblastoma, they can see that from this tertiary structure and quaternary structure, it is a protein binding protein. It can bind lots of other proteins, and it can do lots and lots of different things. Its functions are very, so this is 1986, the gene was cloned. 30 years later, they're still finding out the new things that it can do. It's not really well categorized what it can do. But what it is involved in, the protein is involved in controlling cell division. So it's called a tumor suppressor gene because if you have one copy of this, one copy of the gene producing a functioning protein is enough to keep the control of cell division, keep the constraints so that you don't get this out of control cell division. But once you have no copies of it in a cell due to the mutation events, then those cells will develop into cancer cells. Since retinoblastoma, I think there's over 100 tumor suppressor genes have now been identified. I'll just name a couple of important ones. So the ones you may have heard of because it's in the news and things are I think they call it BRCA, but BRCA-1, and that's on one chromosome. And BRCA-2, that's a different gene on another chromosome. If you have one bad copy of these, then of either of them, then you're much more likely to think you're 10 times more likely to develop breast cancer and up to 100 times more likely to develop ovarian cancer. They're very strongly associated with those diseases. And so there are screening projects, I think, and people who have mutations of those genes get regularly tested for those two kinds of cancers. So you've probably heard about them in the news. They are not so much involved in cell division. Apparently, these are involved in DNA repair. So DNA gets broken, gets damaged. They're involved in repairing it. Again, they're both very complex proteins, like retinoblastoma. They interact, they're protein-binding proteins. They interact with 100 plus different gene products related so far, and their function in particular is not particularly well studied, although they are involved in DNA repair. Another one which you may have heard of is P53. That's the size of, if it was 53,000 molecular weight, this protein, and it's, I think it's because it's mutated in greater than 50% of human cancer cells. Okay, human cancers. So if you look for that in the tumor cells themselves, it's mutated very, very frequently. P53, that is involved in triggering apoptosis, which is programmed cell death. So if a tumor does have very badly damaged DNA, the proteins like P53 and other ones associated with it, again, it's a very complicated procedure. They start a process of the cell where it kills itself, essentially, and macrophages come in and phagocytose and remove all the debris. So there's a few examples of tumor suppressor genes. And just if you remember, these, for them to have that, you could say that they are recessive in sort of Mendelian genetics. Recessive because if you only have one copy of it, they're okay. You're normal. Your phenotype is normal. In order to have this sort of cancer phenotype, you need to have both genes knocked out.